Good afternoon. I'm Chris Fanta. It's my pleasure to welcome you to our Asthma Grand Rounds, which is the first of the 2019-2020 season. And before introducing our speaker, for those who are joining us by live webcasting, I just want to remind you that you can submit a question at any time by texting it to the number shown here, 617-513-6043. And also, if you'd like CME credit, those of you in the audience can sign up at the entrance, and those watching online, just send to the cfanta@partners.org address your name, medical degree, and email address, and we'll award you your CME credit. Great. So I suspect that most of you in the audience already know Dr. Scott Weiss. It's a great pleasure to introduce him. His career spans 40 years, I think, of leading the field of asthma research, beginning as a respiratory epidemiologist and then a geneticist, and now in network medicine, really reflecting or shaping the arc of the Channing laboratories and now the Channing division of network medicine. And he's also the director of Partners Healthcare's Personalized Medicine Initiative. And he's going to talk to us today about a multi-omics approach to lung development and the origins of asthma. It's a great pleasure to have Dr. Scott Weiss.
Thank you, Chris. It's a pleasure to be here. Okay, so disclosures. I write for UpToDate, so I get royalties from UpToDate, and I've been funded by the NIH for 43 years. And most of the stuff that I'm going to talk to you about is related to funding for the VDAART trial, which I'm going to talk about. So what's the outline? So we're going to talk a little bit about when asthma begins, and the message there is it's a lot earlier than you think it is. And I'm prompted to put that in here because when Jeff talked last year, Jeff and Chris got a question from the audience about when asthma began, and they weren't sure what the answer was. So I thought we'd deal with that. How did we decide to study vitamin D? The 3-year and the 6-year outcomes, so intent to treat analyses from VDAART, and then some other analyses that we did. A little bit of a digression about vitamin D genomics because it relates to this issue of when asthma begins. And then I'm going to tell a story about ORMDL3, which is an asthma gene on chromosome 17q, and Sphingolipids and how vitamin D is related to those. So it's a lot to cover in 50 minutes, but we'll zip through it. So when does asthma begin? So first of all, asthma is really a composite of two clinical phenotypes. One is the one on the left, which is disorders, structural changes in the airway, inflammation, remodeling, airways hyperresponsiveness, increased bronchodilator response, which is not exactly the same as airways hyperresponsiveness. I think that people don't recognize sometimes that some of the features that are associated with emphysema, like loss of elastic recoil, are actually features of asthma, even from a relatively early stage. And then the other phenotype is an immune phenotype. So this Th2 type of inflammation that's associated with the disease. And obviously, common disease, a lot of healthcare costs, so people are interested in this. But I think the thing I want you to keep in mind is these two phenotypes, one structural and one immunological, are features of the disease that are actually mirrored in the genetics and develop at different stages and in different ways as the disease manifests itself. So what about diagnosing asthma in early life? Well, this is a huge problem clinically, and the pediatricians who are online listening from MGH know this very well. Symptoms are intermittent, and they're extremely common. Roughly 40% of kids will wheeze during the first 3 years of life, drops to about 20% after age 3. So a large number of kids that are wheezing. And flow in the airway is inversely proportional to the fourth power of the radius. And the reason that's an important relationship is you get very small changes in airway size, and you can have very big changes in terms of symptoms and lung function in a small child. It's really impossible clinically without invasive technologies to distinguish between airway inflammation and just the presence of small airways as the cause for wheezing. And pediatricians in general are reluctant to label children as asthmatic, at least initially when they first see them, because they like to sort of have some sort of experience, since asthma tends to be syndromic and it doesn't have some sine qua non that you can say this is a laboratory test that tells you whether somebody has asthma or not. Children whose symptoms do have them decrease as they grow, because as you get older, the airways get bigger, the lung gets bigger, and the symptoms go away, and hence the 40% prevalence and the 20% prevalence. But these phenotypes don't breed true. If you have low lung function, you can have low lung function without symptoms, and you can have symptoms without low lung function. And there's no really clinically reliable way to predict which children are going to receive the diagnostic label, and the lung function is somewhat uncoupled from the symptoms, because you can't really reliably measure lung function until kids are 4 or 5 years old. So I think this is a really interesting clinical problem. It's actually one that I'd like to apply genomics to see if we could genomically predict on the basis of cord blood or early life blood samples who's going to be asthmatic and who's not. But it does suggest that during the first 3 years of life, you're going to have a bunch of people that are in there whose symptoms are potentially going to go away because they've just got small airways and not some sort of ongoing inflammation. And the big thing that people say is, well, if you wait till age 6, you're going to be better able to say what's going on in terms of the trajectory of the lung function, of the symptoms, and be better able to diagnose who's got asthma. So this is a famous paper by an allergist at the Mayo Clinic who was seeing pretty much everybody in Olmsted County in Minnesota, which is where Mayo is located. And most of the people that he saw, were diagnosed as having asthma before actually the age of 4, let alone the age of 6. But and he was diagnosing a huge number of people in the first year of life. But again, experienced clinician, and maybe he's better off, better able to do this than other people. What was the experience in VDAART? Well, we had a total of 707 of the 806 live births who were followed fully to age 6, so 88%. 133 kids had active asthma at age 6, or 19%. And of these 133, 71 had an asthma diagnosis at age 3. So 53 of the people that had asthma at age 6 were diagnosed before, well before, were diagnosed at age 3. And then among those who were inactive or had no asthma at age 6, most of them did not have asthma before age 3, but there were 37 additional cases that were picked up after age 3. So overall agreement between asthma diagnosis between age 3 and age 6 was 86%, but that's driven largely by this large number of people who didn't have asthma at age 3 and didn't have asthma at age 6. So picked up a few more cases. But this is all going to be important when we start thinking about what were the results of the trial at age 3 and what were the results of the trial at age 6. So how did we decide to study vitamin D? So the guy on the left, who's now the chief of pulmonary at Children's Hospital, came down from Montreal, was sent down to Boston to study here, and he was going to work with Jeff, but Jeff was going to the New England Journal, so then he came over to talk to me. And he said, well, I've been in Montreal and I've been fine mapping chromosome 12q. And I said, oh, I just got a grant to do that. And he said, great. And so he came to the lab as Benji Raby. The first paper that he did was this microsatellite paper that we published in Human Molecular Genetics, where we looked at microsatellite markers in relationship to asthma. And we found linkage on chromosome 12q, very near the centromere here. And then we subsequently went on and fine mapped that region, and we found the vitamin D receptor as a gene for asthma and published that with the group in Montreal at about the same time in the Blue Journal. So that was the first suggestion that this gene, and this was really right after the human genome was mapped. This was like 2004, 2003 and 2004. And so we said, well, let's do some epidemiology and see if we can find this relationship epidemiologically. And so that's where this guy comes in. It's Gus Latonwa. He's now the chief of pediatric pulmonology at the University of Rochester. And we did two studies. The one on the left was done in Aberdeen, Scotland, and the one on the right was done in the Viva study, where we looked at maternal intake of vitamin D in relationship to asthma wheeze in kids. And I think that, I can't remember exactly the definitions here, but I think this was, both were asthma and recurrent wheezing, I think. But really the key point is that the referent quartile in the US study had the same vitamin D intake as the lowest quintile in the Scottish study. So if people in Scotland got almost no vitamin D in their diet. And the bottom line from both of these studies is that they suggested that vitamin D could result in about a 50% reduction in asthma occurrence. If we gave vitamin D to pregnant women, we could potentially reduce asthma by 50%. Seems like a pretty big number, right? I mean, that'd be a pretty big deal. We're spending $80 billion a year on asthma. If we could cut the number in half, you might save $40 billion. That's a lot of money. So anyway, a little bit about vitamin D biochemistry. So 7-deoxycholesterol is in the skin, and when sunlight, UVB, hits the skin, it converts the 7-deoxycholesterol to vitamin D3, shown here, and that's transported to the liver, where it's hydroxylated at the 25 position, and then it's transported to the kidney, where it's hydroxylated at the 1 position, and then it's internalized into the cell, and has two sort of pathways in terms of its effect. One is an endocrine effect, which it's involved in calcium uptake and PTH synthesis, and this is the bone piece of the equation. But the second piece, and the piece that we're talking about, is the autocrine and paracrine functions, where the vitamin D actually trips a cascade of 2,000 and some odd genes, and it is working in the tissues, okay, not in the serum and not through these endocrine mechanisms, to be associated with a variety of immune functions. So this is an example of what the geneticists call pleiotropy. One molecule, multiple effects. We don't measure the 125 in the blood. If you go get your vitamin D levels tested, which I urge everybody here to do, they won't report your 125 level, they'll report your 25 level, because the half-life of the 25 is in weeks, and the half-life of the 125 is in days. So it's the 25 is used as a proxy for the 125. And for what we're going to talk about, we're going to be using the serum levels as a proxy for the tissue levels. And that's a very important concept. The other important point I want to get across here is all of these different types of immune cells, macrophages, dendritic cells, B cells, Tregs, Th1 cells, Th2 cells, all of these cells have the complete immune biochemical machinery so that they can take vitamin D3, remember that's the precursor, or the 25-hydroxy vitamin D, which is the partially active form, and they can convert it into 125-hydroxy vitamin D. So these immune cells, they're sitting in your tissues. They're not sensing the serum level. They're sensing what the level of these biochemical metabolites are in the tissues, and they're responding to that. And the other important point here, this is a fat-soluble vitamin, right? So if you got a lot of excess adipose tissue, that's going to serve as a sink and suck up a lot of the vitamin D. So I think this is almost like the Democrats and the Republicans, because you've got the endocrinologists who are off over here saying, well, the only thing that matters is the serum level. And the only thing that matters in terms of the serum level is what happens with the endocrine effects, the bone effects. And then you have people like me and Gus, who's on the webcast, I think. I got to stay at the microphone. Okay. So Gus is on the webcast, but who think that what really matters for immune diseases is the level in the tissues. And those two groups of scientists aren't talking to each other, or not talking the same language. Final point here about the biochemistry, and then we'll get off this. Pregnancy is a really important immune event. And then there are different immune things that happen at each stage in pregnancy. But the important point that I want to convey to you guys is that vitamin D is active at every single stage here. Vitamin D is important for implantation. It's important for sperm viability. It's important for egg viability. It's important for normal implantation and controlling the inflammatory stage of implantation in the first trimester. It's probably also involved in when the baby decides he or she wants to come out. And it's also important for helping the baby make the transition from the protection of the mother's immune system to developing his or her own adaptive immune system. So promoting the innate and the transfer from innate to adaptive immunity in early fetal life. So a lot of different stages here. Vitamin D is important and critical at every one of these stages during pregnancy. So let's just summarize everything I've told you so far. Vitamin D locus is linked to asthma. Vitamin D intake, not levels, in mothers was linked to asthma wheeze symptoms in 2 studies that Gus and I did. Endocrine effects are linked to serum levels. Immune effects are linked to tissue levels. Relationship between serum levels and tissue levels is not quantitatively determined. And vitamin D mediates all aspects of pregnancy. So, you know, it seems sort of naive now, but, you know, Gus and I knew that 2/3 of the world's population is deficient. 2/3 of the people in this room are probably deficient. And we knew that if we were going to do a study, to try to study this, we were going to have to do a clinical trial where we supplemented people, because you couldn't do an observational study and have it have meaning. But what we didn't know was the exact dose to give or the timing, you know, when in pregnancy this was important. And those 2 things came back to haunt us. But it just gives you, I mean, even just as we're this far into the story, you can see this is complicated stuff here. This is not sort of simple. Okay, so with that background, let's interpret the intent to treat outcomes of the clinical trial. So we designed a clinical trial. We submitted a grant to the clinical trial study section in NHLBI. We got the clinical trial, 4,000 IU plus multivites in the treatment arm, 400 IU in the control arm. NHLBI wanted to limit the cost of this, so they said, we want you to recruit pregnant women that have asthma or allergies in their family, or they have it. We started the trial between 10 and 18 weeks of pregnancy, because that just seemed like the earliest we could possibly do it. We monitored a whole bunch of things shown here in terms of potential complications of the vitamin D, and then we got asthma wheeze outcomes, but we got blood draws at 1 year and 3 years in the kids, and at 1 year in the parents. Now, the first thing we found, now, I would say that we didn't have to go to the FDA to get approval for this 4,000 IU dose, because Bruce Hollis, who was at the University of South Carolina, who's a vitamin D biochemist, had done studies showing that 4,000 IU was safe in pregnant women. So FDA said, fine. There's prior literature that says this dose is safe. Even though the recommended dose is 400, you can go ahead. And the first finding of the trial was this. We undershot in terms of the dose. So what you see here, this is the placebo arm, this is the treatment arm, and this is the 20 nanogram, which is the bone health level that the IOM recommends, and this is the 30 nanograms per ml, which is what we think is the minimum for immune health. And you can see that even with the high dose, only 75% of the women achieved 30 nanograms per ml. So keep that in mind, because it basically says that we undershot with the dose, okay? So bad on us, because we undershot in the dose. And obviously, the point I'm making here is that most of you have levels that are around 20. How many people are taking supplements, vitamin D supplements? One person? 2 people? 3 people? That's about right. Better all go get your vitamin D checked. That's all I can say. We didn't have any complications. There were no serious side effects. This was sort of interesting. Preterm birth was almost statistically significant reduction in the treatment arm. That was not a secondary outcome. And the preeclampsia was the same in both arms, but Human Mirzakhani, who's sitting right up there, wrote a great paper in JCI where he showed that if you looked at the early level of vitamin D, had a big impact on preeclampsia occurrence. So dose was safe. And then this was the main result of the trial at 3 years. And this is a plot of proportion of asthma wheeze at 3 years, Kaplan-Meier plot. And the red line is less asthma wheeze. And you can see here we just missed statistical significance, and we saw a reduction of about 20%. Now, what did I tell you that we saw in the observational data? It's about 50%, right? 50% reduction. So we looked at this, and unfortunately, we're dealing with a journal that said, well, P.05 is statistically significant and P.051 is not. So this ended up getting written up as a negative study. Whether it's truly negative or not, I'll leave that to you. But the reasons we got the results we did at 3 years, I already showed you we didn't give enough vitamin D, but we also didn't give the vitamin D early enough. As I said, vitamin D is important for implantation and maintenance of normal pregnancy. It also influences branching morphogenesis, and I'm going to show you actually some genomic data on that. When does branching morphogenesis occur? It actually occurs in the pseudoglandular stage of lung development, which is between 4 weeks and 15 weeks. What was our window for enrollment? 10 weeks to 18 weeks. So right away, you can see that that baseline level of vitamin D is going to be a critical determinant of what happened, because asthma starts with branching morphogenesis being abnormal. That's when it starts. It starts in the first trimester of pregnancy. Burn that into your memory, because that's a really important thing. The other key point here, and this is a central feature of clinical trials that you need to know, is that nutrient trials are fundamentally different from drug trials. The whole principle of, and the hierarchy of study designs in epidemiology is that clinical trials are at the top, longitudinal observational studies are second, case control studies are third, cross-sectional studies are fourth, and that there's a hierarchy of information related to that. But in nutritional trials, and this is true of every vitamin D trial that's been published in the New England Journal over the last 3 years, they just had one published the other day, there can be contamination of the control group. If you have a drug trial, you have drug in the treatment arm, and what do you have in the control arm? Nada. No drug. Placebo. Nothing. If you're doing a nutrient trial, you've got more vitamin D and you've got less vitamin D. And if you randomize people and for whatever reason, they have a certain amount of vitamin D on board, and you're randomizing at a critical point in time where developmental things are going on that could be related to your outcome, that baseline level of vitamin D could have a big impact, okay? And so this is a fundamental concept about nutritional trials and drug trials and how they are different. Randomization accounts for a multitude of sins in clinical research, but it doesn't always work. And this is a situation where it doesn't work. Okay, so obviously we were disappointed. We felt like we were bozos. We didn't know what we were doing. So we did our best to see if we could salvage what we had here. And so the first thing we did is that we made an alliance with the COPSAC group in Copenhagen, Denmark. And what we did was we did a meta-analysis of their trial, which used the lower dose of vitamin D given later, well into the second trimester, sort of 22 weeks, something like that. And we did a meta-analysis, and that's shown in the upper left-hand corner here. So we did just a meta-analysis because statistically, what's the effect of that misclassification of the baseline vitamin D level? It's just going to reduce your power. So if we increased our sample size, we could potentially achieve statistical significance, and lo and behold, statistical significance with about a 20 to 25% reduction, which is about what we found in our trial with the p.051, but now we're looking at p.02. So for those of you that are p-value aficionados, the meta-analysis clearly shows statistical significance when you combine the two trials, okay? But then we went further. We said, we believe that this initial level is a critical factor because if that level's normal, then it's going to have an influence on branching morphogenesis, and it could be protective, and those people could be in the placebo arm, and so we're getting a reduced effect. So we adjusted for the baseline level over 30 and under 30, and what do we see? When we do that, we get exactly, almost to the second decimal point, the effect that we saw in the observational studies, about a 50% reduction in asthma when we control for the baseline level. Now look over here. This is a very important graph because this is a graph that shows the probability of asthma or recurrent wheezing as a function of the vitamin D level. And this is in nanograms per ml, and 30 nanograms per ml is over here, okay? 40, 50, 60, 70, 80. And what do you see about that line? There's no threshold. It's like a continuous thing. It's more protected. Now, admittedly, we got relatively few people out here with the higher levels, but it suggests that higher levels, certainly 40, 50, 60, would be better than 30. So again, another argument to suggest that Weiss and Latanja screwed up. They gave the wrong dose, and if they had given the 6,000, they would have had more of the people out here, and they would have seen a bigger effect. Okay, but it's 3-year outcomes, and people could say, well, you know, 3-year outcomes, you can't diagnose asthma at 3 years. I would say something else. I would say you can diagnose asthma at 3 years, and what's more, to the extent that there are a bunch of people in there that don't have asthma, what's that effect going to be on the clinical trial? It's going to be a null bias. It's going to make you have less of an effect, not more of an effect. Now, the other thing we did, which is a critical feature of this trial, is that NHLBI didn't give us money to re-randomize the kids after they were born. And that's another deficiency of the feature of this trial, because we were only looking at the effect of the vitamin D on the mother during pregnancy, not the effect of vitamin D on the baby after pregnancy. And what was I saying about the vitamin D and the immune system and being able to help the baby develop normal and adaptive immune system? That piece, the immune system piece, is very plastic. That's not going to go away. The part that we were really influencing was fetal programming of lung development. We were affecting what was actually happening with the structural development of the lung with this particular trial. All right, so we can fight about the p-value, we can fight about the definition of asthma, we can fight about the meta-analysis, plenty of controversy here. 6-year results. So the intent to treat analysis at 6 years for asthma wheeze outcomes was negative. It was 0.05, it was not even close at 6 years. This is an observational analysis where we looked at the average vitamin D, and so again, sort of truth in advertising here. It does show an effect. It does show some improvement if you think about the average, and we adjusted for a whole bunch of covariates, but it's an observational analysis. It's not intent to treat. But this is interesting. This is the lung function data. Lung function was a secondary outcome at 6 years, and what you've got here is airway resistance in this panel and FEV1 over here, and it's stratified by vitamin D levels so that the purple ones are the high vitamin D and the blue ones are the low vitamin D. And what you see here is higher FEV1 and lower airway resistance at 6 years. What's the argument here? Secondary outcome, did a lot of comparisons. Is it really important? Okay, accept that. I accept that as a legitimate criticism, and yet it does suggest that fetal programming could have been altered, and these changes in lung function that we see at 6 years are a direct result of that vitamin D that we gave to the pregnant women during pregnancy. Just a thought, it's a possibility. All right, so let's summarize the 3-year and 6-year results. So we got close to significant results in the intent to treat at 3 years for asthma wheeze. We did get significant results for specific IgE at 3 years. Meta-analysis was significant at 3 years. This effect was greater if the baseline level was taken into account. Vitamin D was safe. It wasn't associated with any untoward side effects. The year 6 results were not significant in the intent to treat. The observational results for asthma wheeze were suggestive, and there was suggestive evidence that there was an effect on airway resistance and FEV1. All right, side digression to talk about asthma genomics. So Kellen and Alvin Coe and I did this study where we got gene expression data for vitamin D using both supervised and unsupervised learning, got this gene set, found a set of common genes. Then we looked at lymphoblastoid cell lines from asthma cases and controls, and vitamin D and not vitamin D treated LCLs. And we looked at the overlap here, and we found a set of vitamin D pathway genes that were affecting lung development in these discarded fetal tissue samples. So this is, don't tell Trump, but this is discarded fetal tissue research. It's actually sanctioned by the NIH on that one. But all of these were from aborted fetuses during the first trimester, when you're in the stage of pseudoglandular. Oh, right. Okay, I gotta stay here because if I don't stay here, nobody can hear me. The stage of branching morphogenesis and pseudoglandular stage of lung development. So exactly the time that we were randomizing people when that initial vitamin D level was taken. Okay, so now you could say, well, that's a sort of interesting story, but you did a clinical trial, it wasn't significant. Is there any other data that would make you think that vitamin D might actually be related to asthma? And that's where we get to this chromosome 17q story. So 17q12 is the first GWAS-identified asthma-susceptibility locus. The second one was identified by us at the Channing PDE4D in a paper that came out a year or two after this. This asthma locus is very, very strong in children. It's less strong in adult. It's actually a 3 or 4 gene locus, but there are 2 major genes, ORMDL3 and gasdermin B. And one of the things that's emerged is that a lot of the environmental factors that influence asthma are transduced through this locus. So cigarette smoke, viral illness, asthma severity, other types of respiratory infections are all sort of transduced through this locus. Mouse studies have shown that if you put the ORMDL3 gene into a mouse and overexpress it, so it's a transgene, you get spontaneous increases in airways hyperresponsiveness. This increase in airways hyperresponsiveness is associated with airway remodeling, peribronchial fibrosis, importantly, without airway inflammation. It turns out, and I don't have this in the talk, but the gene that's driving the inflammation is the gasdermin B. So this gene is driving the remodeling and the airways responsiveness. It's very similar to what we described before when we said we've got structural changes and we've got inflammatory changes. And so David Boyce, who's the person that's done a lot of this work, has got this same sort of idea that there are two primary drivers to the asthma phenotype. So if you do siRNA knockdown of ORMDL3 in human bronchial epithelial cells, it leads to increased sphingolipid metabolites. ORMDL3 inhibits serine palmitoyltransferase, which is the rate-limiting step for the production of sphingolipids. And so ORMDL3 transgene mice have reduced levels of sphingolipids. So now we've got a link between ORMDL3 and asthma and ORMDL3 and sphingolipids. So where's the vitamin D here? That's what we're going to show you. So this was the conclusion of Brody's editorial in the Blue Journal just a few months ago. Although these studies support a role for ORMDL3 in regulating sphingolipids, further studies in humans with asthma are needed to determine whether any of these ORMDL3 regulated sphingolipid pathways contribute to the pathogenesis of asthma. He made one mistake in that sentence. What is it? I don't think you want to be studying prevalent cases. I think you really want to be studying people before they get asthma, and we'll get to why that is. But so we looked at ORMDL3, and what we found is that there are a whole bunch right here of vitamin D receptor binding sites on the gene. So that's an interesting finding in and of itself, that there would be a lot of vitamin D receptor binding sites in this gene. I haven't done this study. It's on my list of things to do. But my suspicion is that if you saturate those vitamin D receptor binding sites, you downregulate the gene. And it doesn't get expressed. And so you get more sphingolipids, and it's protective for asthma. But what we did is that we took this functional SNP, and we did a study in 2009. It's published in the American Journal of Human Genetics to say that this SNP is an insulator, and it actually controls the haplotype ORMDL3 and gasdermin B. So we looked at that SNP, and we looked at stratified by genotype, and we showed that in both VDAART and COPSAC, you had less asthma wheeze if you had the GG or the GC genotype. We didn't just do it in one study, we did it in both studies. So we've got evidence now that in VDAART, that the vitamin D results are being influenced by ORMDL3 genotype. Okay? And here's the network diagram, the biochemical pathway for sphingolipids, and here you've got ORMDL3 blocking serine palmitoyltransferase. And we then went on and said, well, if there is a relationship between genotype and vitamin D response in the trial, maybe there's a relationship between ORMDL3 genotype and sphingolipids, and lo and behold, we showed that here in VDAART, that there's more sphingolipid production in the GG and the GC genotype for most of the sphingolipid metabolites. And then we went on and said, well, maybe vitamin D can increase sphingolipids in bronchial epithelial cells, and if we're right, this effect should be blocked by ORMDL3, and that's what we've shown here. So this is SP1, one of the primary metabolites of sphingolipids, at different concentrations of vitamin D with and without ORMDL3, and showing that, in fact, ORMDL3 does, in fact, inhibit the vitamin D effect on sphingolipids. So here's what is actually going on. Vitamin D actually will increase sphingolipid metabolism and decrease asthma risk. But if ORMDL3 is activated, it will block that vitamin D effect, and you'll get less sphingolipids. And if you get less sphingolipids, you're going to get ineffective branching morphogenesis and increased airways hyperresponsiveness. So let me just summarize what I told you. Chromosome 17 is the most important childhood asthma locus. The locus expression is controlled by VDAART binding sites. Increased VDAART, less ORMDL3 expression. Mouse studies indicate that decreased ORMDL3 expression is associated with less airways hyperresponsiveness. There's an interaction between a functional regulatory SNP and vitamin D treatment on the results in VDAART and in COPSAC. And mouse studies indicate that ORMDL3 is a negative regulator of sphingolipid synthesis. Human integrative genomic studies indicate that the protective genotype is associated with higher sphingolipid levels, and higher sphingolipids during fetal development are protective for asthma. Okay? So now, if you didn't believe the clinical trial stuff, now we've got a tie between the vitamin D and the genetics and a biochemical pathway that is potentially important, that is a novel pathway. Why can't you, we'll come back to this because somebody's going to need to ask me this question because I won't have time to cover it, but somebody should ask me, well, what happens to sphingolipid levels in prevalent asthma cases? So the most important thing I want to tell you is that although my laboratory has been very interested in vitamin D, I don't think that that's the only nutrient that's critical for lung function development. Vitamin D competes competitively with retinoic acid at the RXR receptor. David Schwartz has done studies showing that folate can influence lung function. Hans Bisgaard and the COPSAC people published a paper on N3 fatty acids in the New England Journal and suggested that deficiencies in N3 fatty acids may potentially be important. I have some problems with that paper, but I think that N3 fatty acids plausibly do have a role in asthma development. But I think the most important thing about the vitamin D, and I don't think that the sphingolipid pathway is the only pathway that vitamin D is influencing here. There are at least three others that we're interested in my lab: bile acids, the N3 fatty acids, and the interaction between vitamin D and N3 fatty acids, and the serotonin tryptophan pathway. What is important is that the mechanism by which the vitamin D is acting is both directly and indirectly through the gut microbiome. So one of the most fundamental questions in biology is how human beings tolerize. P.D. Medawar, who's an Indian immunologist, won a Nobel Prize for this in 1996. There are two ways you tolerize. You tolerize through bacterial antigen, primarily from the gut because it's the biggest source of antigen, but you tolerize through these pathways. And if you go and look in the literature, if you go to PubMed, you'll see a bunch of articles about bile acids and N3 fatty acids and sphingolipids and how they influence immune system ontogeny. And all of these things are driving how your Tregs, NKT cells, Th17 cells, how they develop normally. So it's the vitamin D is operating both through direct effects on the biochemistry, but the biochemistry is having a direct effect on immune function. We've actually done a paper with Kathy Sarwar, who's an allergist now on the faculty at the Channing, did a paper with Patrick Brennan showing that the sphingolipids have an important impact on food allergy occurrence in VDAART. So some people in my lab think I'm a vitamin D crazy and it's only vitamin D that I'm interested in. I think vitamin D is monumentally important because so many of you are deficient, and 2/3 of all pregnant women are deficient, and because it has an impact on a wide range of other diseases, preterm birth, preeclampsia, asthma, whatever. But I think there's a predominantly autoimmune disease and asthma is in that category, are being influenced tremendously by what goes on during fetal development, and there's a bunch of different micronutrients that are important in terms of this. So this slide just summarizes all of the articles that we've done over the past 20 years on this. Again, I want to emphasize that VDAART isn't the end-all and be-all. It was high-risk participants, that we got the timing and dose wrong. A baseline level had a big effect. The trial started at 14 weeks, but lung development starts at 4 weeks. Preeclampsia probably starts at implantation. We had a lower proportion of women than expected achieve sufficient levels. We didn't give postnatal supplementation. African Americans, I mean, there's a huge health equity thing here because African Americans have more asthma, have more preeclampsia, have more preterm birth, and they're much more likely to be vitamin D deficient and be severely vitamin D deficient. So I'll finish with this. Nullis et verba is the motto of the British Royal Society. It means not by words alone. So, you don't do science by just talking. You do science by doing studies and publishing them. So I think that pregnant and lactating women are at high risk for deficiency and vitamin D, and deficiency increases in both mother and offspring for these various diseases. Our data and others suggest that a dose of 6,000 is probably necessary to achieve serum levels of 40 nanograms per ml, which we think is probably optimal, and that we need a large pre-pregnancy-based trial with genomics to prevent both preeclampsia and childhood asthma because they're incredibly costly and important with regard to health disparities. A lot of people that worked on this, including Bruce Levy and Benji and Gus, but a lot of the other people here contributed to the data that I showed you, some of which was in our Amberson lecture. The Sphingolipid stuff was just published in the European Respiratory Journal, and the 6-year outcomes are under review at the New England Journal now. So Benji, Gus, Jessica led the metabolomics. Vince Carey, who's been the statistician on VDAART, Amitabh Human, who's here, Rachel Kelly, who was the first author on the Sphingolipid paper, and Xiaobo and Fang, who did the cellular work on the assays. Thank you.
Fantastic. Thank you so much. We welcome your questions. Maybe I'll put up the slide again about those who want to send questions via text. But maybe I could ask, now that we know that asthma begins in utero and is influenced by branching morphogenesis, can you amplify on that? Are we talking airway number, airway diameter, cross-sectional area? What's the impact?
I think now that we know this, I think we could actually get to the actual cells and the morphometric effect. But I think we've narrowed it down to a stage of fetal development and some of the nutritional factors that influence that. And I think that there's a bunch of different studies that could be done in collaboration with neonatologists or lung developmental biologists to actually look at that. I mean, the sad part of this to me is that this story is incredibly complicated and much more complicated than, if knowing what I know now after having been a scientist for 43 years, if I knew how complicated this is, I'm not sure I would have
started earlier.
I don't know, but the point is that I think that I need to write a review article to summarize all of this so that people can understand how complicated it is. And then once that's out there, I think it'll be a little bit better able for people to start to think about it more realistically. I mean, the bottom line is that even though the trial's negative, I think vitamin D is critically related here, and we're onto something that's potentially important. I'm happy to take other questions from people.
Thank you for the presentation. So I actually have a question about the potential cultivating effects between the vitamin D GWAS and the asthma GWAS, because for example, in 2017, when there was a paper published on Pulse Medicine, they used the GWAS data, vitamin D GWAS data from Sunlight Consortium and a Gabriel Consortium for asthma and UK Biobank for asthma. And then they used the maternal randomization analysis to see if there's any genetic causality from vitamin D to asthma.
Yeah, so the question, just to repeat the question, is there's a paper that looked at vitamin D and asthma and used Mendelian randomization. So does everybody know what Mendelian randomization is? So that they used the vitamin D receptor gene and randomized people to two groups, and they didn't show an effect. And I think that it's my belief that Mendelian randomization doesn't work here. And there are at least three problems. One problem is sample size and power, that you need huge numbers, and that paper did not have adequate sample size. The second thing is, go back to the biochemistry, right? This is not just an effect of vitamin D receptor. There are 2,000 downstream genes that are activated as a result of activation. So you're not randomizing those 2,000 genes, you're just randomizing one gene. Now, Aisha Killick, who's a postdoc who's working with me and with Bruce, is doing a bunch of studies on the VDAART knockout mouse. And it's very clear that if you, first of all, the VDAART knockout mouse is viable, it lives. But it does have a very, very clear Th2 immune phenotype. So I'm much more believing the knockout studies than I would believe the Mendelian randomization studies. Other questions?
There was a question texted about the influence of sunlight and whether
So great question. So sunlight, what about sunlight? So the naked mole rat was not a joke. You are all naked mole rats because all of you spend over 90% of your time indoors. And that's true whether you live here or whether you live in Phoenix, Arizona, or whether you live in San Diego, California. So even in the Sun Belt, and Gus and I wrote a nice paper, I think it's a nice paper, in 2008, where we show that basically a variety of things happened to really decrease vitamin D levels in populations. And first it was television. Well, first it was taking away cod liver oil. Then it was television in the 1950s. Then it was air conditioning in the 1960s. Then it was sunscreen in the 70s. And by the time you got to the 80s, everybody was a naked mole rat. So the epidemiologic studies that look at sun exposure in relationship to vitamin D levels are just not very accurate because and people who want to try to mitigate their vitamin D deficiency by spending time in the sun, basically if you live in this latitude where we live, it isn't going to happen for 3/4 of the year. And I think again, there's a lot of other effects here of the vitamin D deficiency, potential for effects on autism, for ADHD, for a lot of other things. So it's really not a great idea for pregnant women to be deficient. And I think that the current recommendations are just woefully inadequate in terms of what people are taking.
Question here.
Yeah.
Combined in two arms of the VDAART study, the control and the intervention with the high-dose vitamin D to one big cohort, then segregated by twin parts, let's say the maternal vitamin D. Would that help you in terms of tracking?
Yeah, so just to repeat the question was if you, instead of doing a intent to treat analysis, you do a cohort-based analysis based on level, would that give you more information about asthma outcomes? So we have a paper that's being written on that, and there is a Human is actually, and Mindy Liu are actually working on that. And if you do that, you know, where you definitely see a fairly strong relationship between the levels in pregnancy and the asthma wheeze outcomes in the kids. But again, you know, the argument is, well, that's not a randomized controlled trial. You did a different analysis. Yeah, I mean, I think that what it does is, and the reason you're going to get different results than what you would get in a free living cohort analysis is that in a free living cohort analysis, so many people would be deficient that you just wouldn't have any power to see anything. You wouldn't see anything, because if all the levels are low, you have no variation and you have no, whereas if you've given them supplements, you're going to get a broader range and higher levels. You're not going to find many people with levels of 50 nanograms per ml in a free living population in the United States. It just doesn't happen. The other feature here that I think is important is that this is an integrative omics analysis. We combined genetics with biochemistry, the metabolomics, to do an integrative analysis. So I think that one of the things we're very interested in the Channing is different ways of combining different types of omics data to try to get bigger insights into what's actually going on. Anybody else on that?
I don't think, no, but I have one. I don't think we can let you go without the question that you asked us to pose to you about the role of sphingolipids in early childhood, and what are they doing in the airway epithelial cells?
And what about, so thank you. So Chris reminded me that I raised this question, and I think it has to do with what epidemiologists call incidence prevalence bias. If you take prevalent asthmatics and people who already have asthma, and you stimulate them with an pro-inflammatory stimulus, like a viral illness or particulate air pollution or something like that, what will happen is their airway will produce more sphingolipids. And so people would say, well, in the prevalent case, you've got somebody who's asthmatic already, and you give them this stimulus, and they produce more sphingolipids. Sphingolipids bad.
Doesn't fit with your hypothesis, right?
But I think it's a injury repair response, a primitive injury repair response to for abnormally developed airway in a prevalent case. And I think that, you know, probably one of the most important lessons I've learned as being a researcher for 43 years is that you can screw up these causal relationships if you're studying cross-sectional data, because you don't know which way the causal arrow is going. Is it those sphingolipids are trying to be protective, or are they trying to be risk? You can't tell. So, I mean, if you want to ask a really complicated question here, we were looking at maternal genotype and maternal sphingolipid levels here. So somebody could say to me, well, Scott, I want to know how those maternal genotypes and maternal sphingolipid levels are interacting with the sphingolipid levels in the genotype of the baby. And that's a great question. It's a complicated question because you're not going to get fetal in utero sphingolipid levels. But you can measure the genotype of the child and look at that in relationship to the mother, and we do have subsequent sphingolipid levels in the child. So that's another project that we should do to try to think about. But the interaction between the child's genotype and the mother's genotype is obviously a very, very complicated phenomenon. And I think it would be unfair to say that just the genotype of this one SNP is enough to characterize all of the genetic variation in the sphingolipid pathway. We need to do more about looking at other genes that could be, I mean, what's critical about this one, which is, I think, unique, is that you're cutting off production right at the start of the pathway. Whereas if you look at the FADS gene for fatty acids, that's further down the pathway at a point where you've got branching between eicosanoids and leukotrienes. So it's a different place in the biochemical pathway. So I think the other point here could be that you can't biochemistry. I mean, what different kinds of data did I use in this presentation? I used genetics, I used genomics, I used asthma natural history, I used evolutionary history, I used metabolomics, I used integrative omics, I used a whole bunch of different things. But I don't think you can point to one discipline and just say, oh, I'm a biochemist, I just know the biochemistry, and that's going to tell me everything. I think things are too complicated for that. I think you got to have a much more holistic view and be able to take from a variety of different disciplines if you want to try to figure out what's going on.
Not a question, but just one comment that the historical origins of the term Sphingolipid is from the Sphinx and being mysterious and difficult question to answer what they do about. That is playing out very well right here.
Yeah, I mean, I would urge people that are interested in the Sphingolipids to actually think about what's going on in type 1 diabetes. There's a bunch of other autoimmune diseases that are very interested in Sphingolipid metabolism and its potential for, and a lot of stuff in the immunologic literature about its potential importance.
It's a good time to wrap it up. Thank you so much for a fabulous discussion.
All right,
Great.
good.
Thank you.